Alright guys, so today we're going to start our new unit, and our new unit is going to be all about genetics. So we've just finished up with the concepts of cell division, mitosis and meiosis, and when we learned about meiosis or meiosis, we know that sex cells are created that have half the amount of chromosomes or genetic information than an adult. And so as we move forward into genetics, we're going to see how we get our traits from the inheritance of this DNA from our parents. And so what is genetics? Well, it's the study of heredity. And what is heredity? Well, it's the passing of traits from parents to offspring. And just make sure we understand a trait, that's just any characteristic that can be passed down from the parents to the offspring. So essentially when we're studying genetics, we're studying how traits are passed down to the babies. And so we do have a little bit of background info. This is common language in today's world. We hear terms like genes and chromosomes, and especially with advances in DNA and DNA technologies, like these are things that we could potentially hear in our daily lives. And so words like genes and chromosomes are kind of second nature. I think a lot of times we may not know the specific definitions, but we've at least heard of them. And so in our past lessons, we learned that chromosomes were condensed forms of DNA, and we know that the chromosomes are visible during our cell division stages, and the chromosomes contain genes. And so essentially a gene is a segment of DNA that is on the chromosome, and then the genes determine our traits. And it's the chromosomes that our parents are passing down to us. We got half of our chromosomes from mom and half of our chromosomes from dad. In our lesson on meiosis, we learned about homologous chromosomes. Remember that mom and dad both give you a chromosome number 1. And mom and dad both give you a chromosome number 14. And we know that they're homologous because they're the same size and they have the same genes. And those genes are in the same places, known as loci. But we also know that those chromosomes are not identical. Our moms and dads are not genetically identical. And so, once again, we have the same genes, but they're not identical because they can be different forms of those genes. And a lot of this is what we're going to be learning in today's lesson. Now, it can be confusing, or not necessarily confusing, but sometimes surprising, because words like this are kind of common knowledge to us. But there was a time in history where we didn't understand how traits were passed from parents to offspring. We didn't fully understand that mom and dad each gave us half. We didn't fully understand processes like meiosis. And so when it comes to studying inheritance, probably the first person who gave us pretty much all the basics of inheritance is Gregor Mendel. And what's kind of shocking about Gregor Mendel to me is that he did all of this without our knowledge of DNA and without our knowledge of processes like meiosis. Those were not completely understood. The structure of DNA was not discovered until the 1950s, and Gregor Mendel did his work in the 1800s. And so he did all of this inheritance work without the knowledge of the molecule responsible. And I find that pretty fascinating. He also didn't really get credit in his lifetime. People rediscovered his work after he had passed away. But essentially, we consider him the father of genetics because he did lay the foundation for almost the basic ways of how our traits are passed from parents to offspring. Gregor Mendel kind of created the basics. And he developed these 3 laws of inheritance that we're going to take a look at. And so just a little bit of background about Mendel. He was a monk. He lived in a monastery, but he had a scientific mind. And one of his jobs at the monastery was to tend the gardens. And so he just kind of made some observations, and he noticed that the offspring plants usually were similar to the parent plants. And so he kind of started to conduct his own experiment. And he studied over 28,000 different pea plants. And what he concluded throughout these studies was that these factors, which that's the term that he used. Today we use the term genes. But he concluded that these plants transmitted factors from the parents to the offspring. And so what he did was he specifically focused on garden peas. These are the basic little green peas like you might eat with your meals at home or in a soup or something. And these little garden peas were easy to cross, and there were 7 basic traits that were easily observable in our peas. Now, this is mentioned somewhere, but one of the things I want to let you know about is that Mendel really got kind of lucky. Not every set of traits is this or that, okay? So if we go back, you can see that every single one of these had 2 possibilities. The flowers were purple or white. The flowers were axial or terminal. The seeds were round or wrinkled. Notice every single one of these traits has 2 options only. And that's one way that Mendel got lucky. We'll talk about another way as we go through the lesson. And so in order to conduct his test, pea plants have the ability to self-pollinate. And so for flowers, they can self-pollinate when they have both the male and female flower parts. So these are the anthers right here, these little orange things. And this is where the pollen is produced. And then the pollen has to land on the sticky stigma, and then it will go down and fertilize the eggs. And that's what creates the seeds of the plant. And so what Mendel could do is he could remove the male part. He could cut those little anthers and stamen off. And what that would do is that would allow him to physically transfer the pollen between 2 plants. Okay, so he was able to control which plants he wanted to cross. And so what he would do is he went through the same series of tests for each of the 7 characteristics. Okay, so we saw those in the last slide. And he always started with what he called true breeding plants. Now this is what we would today call either a purebred, or we would refer to them as homozygous. So Mendel didn't have this vocabulary. This is what we use today. But essentially he started with pure or true breeding plants. And so what that means is if they're pure, they only produce the same thing. Like if you're breeding dogs and you have a golden retriever, if it's a pure golden retriever, it's only going to produce golden retriever babies when you mate them. So that's what he was dealing with these pea plants. So if they were tall, they always produced tall plants. If they were green pods, they only produced green pods. And like I said, today we're going to refer to that as homozygous. But what he would do is he would take each of these contrasting characters and he would cross a green pod with a yellow pod. He would cross a pure tall with a pure dwarf, a pure purple with a pure white. And so once again, he got lucky. Okay, so he got lucky that every trait only had 2 options. He also got lucky that every trait was on a different chromosome and followed the same type of inheritance pattern. And so what Mendel did was he went through this series of crosses for each of those 7 traits. And so he would always start with these pure or true breeding plants of opposite trait, so purple and white, axial and terminal, yellow and green, etc. And he would cross them. And this is what we would call the pea generation, these original plants, okay, the purple and white, the yellow and green, these were our pea generation. And so this process is called hybridization, because anytime you cross organisms with different traits, we call that hybridization. And so the babies that resulted from this cross are known as the F1 generation. And so all of these are going to be hybrids. You cross 2 purebreds, all of the babies are going to be hybrids, which is what we call heterozygous. Now, one of the surprising things is that the babies only came out showing one of the 2 traits. And this is the trait that we consider the dominant one. So then what he would do is he would take the F1 generation and he would let them self-pollinate. So essentially he would cross an F1 hybrid with an F1 hybrid. And this would produce the babies known as the F2 generation. And here's where the numbers kind of started to show a pattern for sure, is that almost every single time we would see both traits, okay? There would be purple and white flowers. There would be yellow and green seeds. There would be tall and short plants. But it was almost always in a ratio of about 3 to 1. And so you can see here, it's not a perfect ratio, but in almost every circumstance, it was about a 3-to-1. And so it was from these crosses that we get Mendel's 3 laws, okay, that allow us to kind of lay the foundation for patterns of inheritance. And so the first law is the law of dominance, that some factors, this is the term that Mendel used, we use the word alleles, but that some factors were dominant and could mask over others. And this was represented in that F1 generation, the fact that all of the F1 offspring showed only one trait. The second one is the law of segregation, and essentially this has to do with the process of meiosis. So we know that you have 2 alleles, okay? You got 2 copies of each gene, one from mom and one from dad. And what this is talking about is the fact that the parent had 2 factors, but they only gave the babies 1. And so the separation of those alleles had to happen whenever they formed their gametes. And so gamete formation is meiosis, okay? So essentially, mom and dad each have 2, but they only give 1 to their babies. That separation occurs whenever they made their egg and sperm. And so this ensures that we get 1 factor from each parent. And then the third law, the law of independent assortment, and we actually saw this independent assortment whenever we were doing meiosis, but that says that the alleles separate independently from each other. And really this is about different chromosomes. So what happened at chromosome 1 didn't affect chromosome 2 or 5 or 22, okay? So if a trait was inherited on chromosome number 1, that had no effect on traits that were inherited on chromosome number 4, okay? So an example of this is just because you have curly hair doesn't mean you have to have blue eyes, okay? Or just because you're tall doesn't mean you have to have freckles. All right, each trait is inherited independently of the others. And really this has to be on different chromosomes. And so from this work, from these laws, these are the terms that we use today. And really, as we go through the rest of genetics and we do problems like Punnett squares, we need to be very, very familiar with these terms. And so we're going to start with genes and alleles. Now, genes are the factors that control our traits. And essentially, these are specific pieces of DNA that code for our proteins. And our genes are found on our chromosomes. So there's genes for hair color, eye color, height. For the pea plants, there were genes for seed shape, plant height, things like that. Now, those genes can come in different forms, and this is why the homologous chromosomes are not identical, because there could be the dominant allele, there could be the recessive allele, there could be a blue-eyed allele, a brown-eyed allele. So it's one gene, but it's 2 different colors. And so these are my different alleles, my different forms. And so the alleles are what we inherit from our parents. Now, some terms that describe those alleles are either dominant or recessive. And like we saw with the law of dominance, the dominant allele can cover over or mask the recessive allele, and then the recessive allele can be masked over by the dominant. And so here's the thing, if you have a dominant trait, you have at least 1 dominant allele, because as long as there is 1 present, you will see the dominant trait. And this is why all of those F1s only showed the dominant trait, okay? They all had the recessive allele, but it got covered over. And so the only way that you can see the recessive trait is if you get the recessive allele from both parents, okay? So for the dominant trait, you only need 1 dominant allele. In order to see the recessive trait, you must have 2 recessive alleles, only recessive alleles, because anytime there's a dominant one present, it's going to mask over the recessive. And so we represent dominant with a capital letter, and we represent the recessive allele with a lowercase letter. So some common dominant traits that we see in humans, and some of these are a little bit iffy, but the main reason I wanted to show you this is because of like this one right here. I don't want you to think that just because something is dominant that it's automatically going to be the most common. Having 6 fingers and toes is more common. Having type O blood, which is recessive, is the most common. So just because you're dominant does not mean that is the most common trait in a population. But you could kind of do a little quick survey of yourself and figure out which trait you have and whether you have the dominant or the recessive. All right, now some other terms that we use today that came from Mendel's work are genotype and phenotype. Now the genotype are the genes or the allele combinations. And so essentially these are our letter combos. Remember, we always get one from mom and one from dad. So we have two capital letters, that would be two dominant alleles. We have capital and lowercase, that would be dominant and recessive, or we could get two recessive alleles. So the letter combos or the allele combinations are our actual genetic makeup. Now the genotype is going to determine the phenotype, which is what we physically see. Okay, this is the physical trait that we have. So you can remember that phenotype is the physical trait or the physical appearance. So you could have blue eyes, brown eyes, green eyes. The flowers could be purple or white. The plants could be tall or dwarf. You could have the cleft chin or no cleft chin. All of those are examples of phenotypes. Now, we describe our genotypes with the terms homozygous or heterozygous. And the prefix homo means the same, and the prefix hetero means different. And so if you have two of the same alleles for a trait, those are both described as homozygous, so that can be dominant or recessive. And then if you have different alleles, one capital, one lowercase, that is described as heterozygous. And so when we put it all together, the genotype or the letter combinations, we describe those as either homozygous or heterozygous. Remember that capital are dominant, lowercase are recessive, and then the genotype determines the phenotype. So if you are homozygous dominant or heterozygous, notice that both of these are the dominant trait, and the only way you get the recessive trait is if you have two recessive alleles. Okay. All right, so everything we've looked at so far was the work of Mendel, and in all of his, like I mentioned, he got lucky. All of those traits were controlled by one gene with two alleles, and every single option only had two phenotypes. But that's not the case for all of our traits. Okay, we don't just have two skin colors, we don't just have two heights, we don't just have two eye colors. And so not every gene or every trait have a simple inheritance pattern, okay? Not all of them are just purely dominant or recessive. Not all of them are controlled by only one gene. And so what happens is we get some of these other inheritance patterns. And oftentimes we refer to these as non-Mendelian inheritance. And so here's a great review of everything that we're about to cover. So we're going to take a look at incomplete dominance, where it's not just normal dominant recessive. We're going to take a look at codominance. We're going to take a look at what happens if you have more than two possible alleles. And then we're going to take a look at what happens when you have multiple genes controlling a trait. And so we're going to start with incomplete dominance. Now in incomplete dominance, it's just like what it says. The dominant allele does not completely dominate or mask over the recessive. It just kind of covers over. And so what happens is we get a blending of traits, okay? Incomplete leads to a blending of traits, very important. And so some examples of this are like in flowers. The red flower may not completely cover over the white. And so when you get a heterozygote here, instead of seeing just the dominant trait, we see a blend, which would be pink. You could get wavy hair in dogs if you crossed a straight and a curly. You could get medium fur in rabbits if you crossed a short and a long. And so what you're seeing is a blend, okay? Now another type of non-Mendelian inheritance is codominance, and this is where both alleles are dominant. And so neither one is going to be overshadowed by the other. So we're not going to see a blend anymore. Instead, we are physically going to see both, okay? So in codominance, you see both of them because both of them are dominant and both of them are going to show. So some examples of this are roan cattle. You cross a red with a white and you get the combo of roan. You can see both the red and white splotches. If you cross black and white chickens, sometimes that can produce speckled chickens, which are black and white. Another example is the AB blood type. A is dominant, B is dominant, and so if you get both, you physically see both. Another type of inheritance that may not meet Mendel's ratios is if there's more than two possible alleles. So once again, with Mendel, every single one of those pea plant traits had one dominant, one recessive, and that was it. But in a lot of traits, there's more than just two possible alleles. And the more possible alleles you have, the more possibilities you have. So what happens is, by increasing the number of alleles, you increase the genotypic and phenotypic possibilities. It just gives you more variety. Now, just because there's more than two possible, remember that an individual will still only get two, one from mom and one from dad. And we see this in our blood typing. There's 3 possible blood typing alleles, and that gives us 6 different genotypes and 4 different phenotypes, okay? This is also something that you can see in rabbit fur color. So there's 4 different alleles here, and then that gives you different phenotypes. So all this does is increase the varieties. And then you have polygenic traits, and these are traits that are controlled by more than one gene. And a lot of our traits, a lot of human traits are polygenic, and these are going to be harder to predict because it's not just what alleles you get from one gene, but the alleles that you get from a variety of genes. And so once again, we get a wide range of phenotypes that normally looks like a spectrum, okay? Height is an example. You have the shortest to the tallest and every height in between. You have lightest to darkest and all different colors in between. So normally what you see with polygenic traits is a spectrum or a range, okay? So this is an example of skin color. You can see you have very, very fair skin all the way to darker skin and many different shades in between. And then the more genes that control it, kind of the more varieties that you're going to have. And then the last thing is that your genes are not the only thing that determine our traits, okay? Many of our traits are affected by the environment, and we call these multifactorial, okay? So these are the traits that can be influenced by the environment. So like, I'm fair-skinned, but if I lay out in the sun all summer, my skin does get darker. Now, something you need to understand is that I cannot pass that down. That does not change my genotype. Okay, my genotype is still the same. I still have lighter alleles, but I can physically change the physical trait. Okay, height and weight are both examples of multifactorial traits. You know, genetically, I'm supposed to be a certain height, and genetically, I'm probably supposed to be a certain weight, but how I eat, my nutrition, as a child, if someone does not get proper nutrition, they will probably not reach their full height potential. Weight, diet, and exercise are big components to how much you weigh. You can also see this in gene expression in animals and plants. So like rabbit coloring can vary depending on the temperature and whether or not the gene gets turned on. Light can do the same thing for plants. And so the big takeaway here is that it's not just your genetics for some of our traits. Some of them can be influenced by the environment as well. And that is going to wrap up our first lesson on genetics. We will continue and learn some more. Starting tomorrow, you're going to start learning how to do Punnett squares. And it's very essential that you understand the vocabulary that goes along with this lesson, especially those pairs like genes and alleles, genotype, phenotype, homozygous, heterozygous. I hope you guys are doing well. If you have any questions, feel free to reach out, and I hope to talk to you soon.